thank everyone for joining us for this final presentation of the day, at least in the post-approval and generics group. We've got Brett Howe, president of DILIsym Services Division, who will be giving us a nice talk that segues very well from the poster presentations we just had. With that, Brett, your video's up, your presentation's good to go, and I believe audio should be working as well.
Okay, thank you, Ryan. Thank you, everybody, again for sticking around, closing out the day here. I know it's been a long day of presentations and yesterday as well, but we really much appreciate everyone attending and participating in our first ever MIDD Plus conference. So I'm excited to give this presentation. This is kind of an interesting use case, I would say, for the DILIsym platform, and some interesting regulatory interactions and aspects to this as well. You know, we at Simulations Plus have a variety of specialties when it comes to mechanistic modeling and areas of focus. But today I'll highlight really the DILIsym software, which if you were on previously, you just heard from Diane Longo. She had extended a PBPK model that we had developed in-house, developed by Inger to liver injury prediction. Now this is another use case for the DILIsym platform. So today I'll talk a little bit about the model itself and DILIsym and how it's come to be and how it's applied generally. And then I'll shift gears and talk about a specific case study for acetaminophen where carcinogenicity is in question and being discussed and assessed for this compound. And I'll show some FDA correspondence which suggests that the conclusions that we reached with the DILIsym platform were in agreement with those that the FDA reached as well in the process. So the DILIsym platform, we are currently on the 8th release of the software. It predicts and also helps explain drug-induced liver injury, most usually from small molecules. We are moving more towards large molecules as of late. And we're heading towards our next release very soon, which will be DILIsym version 9. And practically, how do we typically use the DILIsym platform or companies that we engage with? Well, predicting DILI liabilities in advance is an obvious one. You know, predict and avoid, and so save money is pretty clear. One of the main goals. But also looking at relative potencies and effects and liver effects. So you can take like molecules or competing molecules within a pipeline and look at those compared to one another. But the last one here highlighted is the one we'll kind of look at in this talk, and that is communicating with regulators on safety issues. Sometimes it's related to requests that come to drug development teams from regulators, and sometimes it's really drug development teams being more proactive and working in this investigative toxicology space to de-risk and to explain in advance of serious regulatory actions being taken. So the general idea with the DILIsym platform is that we've constructed a mathematical model, a fundamental representation of how the liver functions, the biochemistry, physiology, the turnover of the cells. And so that model in of itself is sort of free of drug. It's really focused on liver and liver function. And then we add to that the ability to predict how much drug would be in the liver at a given point in time. And for that, of course, we use our favorite platform, GastroPlus. And then lastly, we do an assessment of how particular pathways or mechanisms might be perturbed by a drug and how that can interact with the exposure and the pathways to lead to a response in a human being or an animal. Now, the DILIsym platform has not been developed in a vacuum. It's been developed within the confines of a consortium called the DILIsym Initiative. This is a partnership between the companies you see here and others currently in the initiative. They have supported financially the development of this software over a period of 10, 11 years, but also have brought intellectual capital and invested time and energy in helping us understand and steer the right direction for this platform. It's afforded us a great scientific advisory board led by Dr. Paul Watkins. It's allowed us to have and formalize some interactions with regulatory agencies as well. So it's been a successful development effort. As a part of the initiative, we have focused on dissemination of information, and you can see some of our information here that we published in 2020, some of which has been presented at this conference this week. But there are a number of good publications you can go and look at that describe DILIsym, talk about the work that we've done for different drugs and different applications. And, just a brief sort of, plea for members. We'd love to have more companies joining. There are a lot of benefits to joining the DILIsym initiative and accessing this platform. There are practical software related benefits, which are listed here, which I could go on and on about. It's a very great value to join DILIsym and RENAsym, the combination of those two. But there are intangible benefits as well that are really great. So to get into one level deeper in terms of what the software is, what it's made up of, DILIsym represents mice, rats, humans, and dogs in terms of the species covered. We have 3 different zones of the liver covered, so some level of detail in terms of liver function and liver toxicity. And we've tested over 80 different compounds for liver injury, where we can assess our predictions compared to clinical data. And our success rate is around 80%. So, 4 out of 5 times the software is predicting well. And interestingly, in many of those cases where it didn't predict well, we've learned a lot as well about what mechanisms may not be involved or about what we may be missing. And we continue to refine not only our model, but also the assays involved in feeding into the model to try and improve that prediction rate. We can do single and combination therapies. We can also, produce biomarkers and outputs. And one example of that is shown here. This is the eDish plot, FDA's way of looking at ALT versus bilirubin and the combination of those two. And generally, you want to stay down in the safe quadrant and not up in the Hy's Law quadrant. So, you can really produce relevant clinically oriented outcomes of the model with the push of a button. Briefly digress and highlight here that we are releasing a brand new version of DILIsym in the very near future. It's a completely redesigned platform and a new software language, much faster, much easier, and better to use. So consider joining our initiative or licensing the platform and picking up a copy. In terms of the process, how you use the software, typically it involves 3 steps. One is to assess the potency of a compound against certain pathways. And we do that by engaging with laboratories to collect data. So a lot of times our projects will include us overseeing and designing studies that are then collected by others, and the data is fed back to us. And that data is related to key pathways like oxidative stress, disruption of mitochondrial function directly, or transporter inhibition. Next, we'll develop a representation of how the drug, how much drug is in the liver and when, the kinetics of that through PBPK modeling with GastroPlus. And lastly, we'll decide how much do we want to dose and what's the frequency, and are these patients fed or fasted, and what type of patients are we interested in. And once we have all of that sort of trial design information put into the software, we're able to make a prediction of risk. In many cases, that's sort of the use case. We're putting these things together, we're making a prospective prediction. In this particular case, for this talk today, though, we're actually talking about a scenario where we had a model of a compound that would be acetaminophen or Tylenol or paracetamol, depending on how you refer to it, very commonly used, analgesic. And we had a model, have a model that's very well validated that describes how acetaminophen can cause oxidative stress, which can lead to liver injury. And so this was more of a case of using an existing model to answer a key question. So let's talk a little bit more about this specific application. It really falls into that bucket of sort of post-approval, hence the session, and also that bucket of analyzing mechanisms, trying to get down into mechanisms and investigative toxicology and tying that back into a particular risk question. In this case, acetaminophen is under review by the California Office of Health and Human Services, and really they're interested in understanding, does acetaminophen cause liver cancer? Now, acetaminophen has a long history of safe uses at therapeutic doses, but there is a history of liver injury, more acute liver injury, being caused at very high doses. And so it was just a year or two ago that the California OEHA called for a review of the carcinogenicity of acetaminophen, we were part of an integrated group of different individuals commissioned by what's known as the Consumer Healthcare Products Association to pull together data and studies to address the question of whether acetaminophen is carcinogenic or not. And we our results were submitted as part of a sort of integrated weight of evidence assessment to OEHA by CHPA. There was a specific supplement for the DILIsym modeling results, which I'll present some of which today. And then there was also an integrated document that referred to those results. And one thing that was nice is in that process, we also published this work in regulatory toxicology and pharmacology as well. So this, more details are certainly available to read in these documents, after the presentation. So the effort really started, and the documents really were framed initially with the validation as the focus. So the question is, if we're going to use this model and submit results to regulators, why should they believe this model? Why should they think that we've represented the acetaminophen processes well enough to then turn around and use this for a regulatory purpose? And so this is a table that just shows some of the areas where we validated the DILIsym model with respect to mechanisms for acetaminophen. So we've heavily validated the model for acetaminophen's ADME and PK with our PBPK representation for its glutathione depletion, the liver injury outcomes, both at the liver level, but also from a biomarkers perspective, the oxidative stress levels that are produced in the liver, general mitochondrial dysfunction and inhibition that occurs because of that, and also protein adduct formation. So these are some of the areas that we documented. And in terms of the mechanism, you can see the figure here. Many compounds that we work with have a sort of multifactorial mechanism. They may hit transporters, they may cause direct mitochondrial effects and oxidative stress. Really, the pathway that we're focused on for acetaminophen is a reactive metabolite leading to oxidative stress, which leads to the reduction in ATP and ultimately cell death. So we submitted results for validation of the model. This is an example of some data in mice where what we're seeing is a depletion of liver glutathione and a nice concurrence between that and the increase in the number of protein adducts. And so what this is really suggesting is that you have a flux of reactive metabolite being produced in the liver. For a while, you have sufficient buffer. These mice have sufficient buffer. But then once that buffer is depleted, you start hitting other proteins, and you have general conjugated proteins showing up in the cell instead of just glutathione being depleted. And what we see here in the sort of the inlay is that with the DILIsym model, we're able to show a very similar trend of depleted glutathione along with increases in protein adducts. So that's sort of at the mechanistic, and almost ADME level validation of the model. There's a number of other validation plots shown here. So this is in humans. We had a wealth of PK data to validate our PBPK representation at different doses. This is 4 grams per day as an example. We had biomarker results. This is plasma ALT or liver enzymes, and the results of this clinical study, and then our simulation results as well, which were very much matched up with very minimal or no ALT elevations at these very low doses. And then we also have fundamental information on how fast the liver regenerates after an injury event. And we have that from both liver injury events with acetaminophen, where they've done biopsies, but also from liver resection surgeries. And you can see with our simulation results with various degrees of injury that our rates of recovery and the rates of regeneration in the model are consistent with the data there. So there's a lot of other data that went into this validation, but the most important thing is that we framed the use case for this with the context of this model is validated for this drug, very heavily. And I think that was an important part of the work. So then we kind of turned our attention to the application. What aspects of acetaminophen's toxicity are we looking at, and how does it relate to carcinogenicity? Well, this is a figure showing a time course of acetaminophen being given to a simulated human, a single average individual, if you will, at different doses. So there's therapeutic at the top and an overdose case at the bottom. And so we have 20 grams per day at the bottom, and then a therapeutic dose at the top of just 4 grams per day. And what we see is that therapeutic dose, no issue. The glutathione, which is in green, is not depleted. There's no serious liver injury at all. We move to the bottom. It's an interesting picture of glutathione depleting and almost in sync with the glutathione depletion is an increase in protein adducts, an increase in the impairment of the mitochondria, an increase in oxidative stress, which is the black line, and then subsequently just after a drop in the number of viable liver cells. And so what we noticed, and really where we were providing input on this whole question, was there's a sharp transition from the safe space of acetaminophen to the unsafe space. And really what you would look for in a drug that causes, carcinogenicity is a sustained low level damage. So a situation like NAFLD and NASH, which leads to, long-term liver cirrhosis, which you can also get hepatocellular carcinoma, later after that in subsequent line of events. In this case, this is really an acute effect. You have, you're either okay or you're not. And that was sort of the point of a lot of these analyses. You can also frame that same question with respect to a dose response. So this is, again, in an average individual in a particularly sensitive region of the liver, the centrolobular region. We've got a dose response, and we see, again, the sharp depletion of glutathione as you increase dose. At the therapeutic doses, plenty of glutathione. We start to get to 10 grams, and now we've depleted our glutathione, and now all of our injury indicators start to go up. So we see a pretty sharp change there. So the dose response suggests that, perhaps APAP has a built-in mechanism against carcinogenicity. It's almost as if the molecule has been designed to either be safe or, when the doses increase, to be, very toxic. And as a result, you don't have these sort of low-level toxicity scenarios where you could get this long-term buildup of toxicity, of DNA damage, of protein adducts, and things that could cause carcinogenicity. So that's what the results at the single individual level seem to suggest to us, and really making a mechanistic argument there. So we looked at the population level, said, well, what about people who might be vulnerable, people who may have lower levels of glutathione depletion or concentration. And so this is a busy figure, but if you bear with me, at the top, we see with the therapeutic doses of acetaminophen of 4 grams per day, see a whole wide range of individuals with different starting glutathione concentrations. And notice that across all of those starting concentrations, which range from, sort of very, very healthy, individual with lots of antioxidant capacity to individuals with 50% depleted glutathione, all of them have sufficient buffer at the therapeutic dose to avoid major liver injury. You have a small amount of mitochondrial effect, a small amount of liver cell loss, but nothing clinically significant. When we go to 12 grams per day, so if we, by a couple days in, you're up to 25, 24 grams of acetaminophen, what we see is we see some of these individuals with lower glutathione starting to have depletion. At the same time, impairment of mitochondrial function, which is going up in red, and then shortly thereafter, a drop in their vital hepatocytes. And so we have, again, this sequence of events. And if you look at it on an individual level, there are individuals who aren't as susceptible. They don't have sufficient glutathione depletion. They have no injury. But you have other individuals who are more susceptible. But the point is, you're either in the safe zone, you have sufficient glutathione stores, or you're not, and you have significant injury. There's not a zone in the middle where you would have this smoldering stress in the liver for many years after taking acetaminophen and see some issue with carcinogenicity. And we converted this to sort of more of a visibly satisfying figure where we've got different regions representing the variability in the population. So we see this region of glutathione depletion in green, where as you go up in dose, you're starting to see individuals with depletion at 10 grams and all the way out to 25 grams. And we see the regions match pretty well as you go through. So the mitochondrial dysfunction region starts to go up right about the time when the glutathione starts to get really low in the first, for the most susceptible individuals in the population, which would be sort of the left side of the figure. And then the fraction viable starts to go down again as you get to the point where mitochondrial impairment's up and glutathione's down. So it shows you this nice sequential step of glutathione down, mitochondrial dysfunction occurring, liver cell death occurring, and again points to the sort of almost bimodal nature of this particular effect. And we can see also denoted here are the percent of patients from clinical data who had, patients up to this dose X who had AST elevation greater than 3 times the upper limit of normal, and here who had bilirubin elevations. And really, it's really nice to see that the clinical data shows a dose response and an increase in these biomarkers that is quite commiserate with our region of fraction viable starting to go down about 20 grams. So it's almost right around 15 to 20 grams. You start to see this effect and start to see the injury be more significant and give you more biomarker elevations in the population. So when we looked at this figure, we looked at this figure on the left and sort of analyzed this mechanism. What we concluded was that the simulations that include the population variability really support that across a wide array of patients, across a wide array of doses, acetaminophen really results in significant oxidative stress or DNA effects under conditions that cause cell death. So you're not going to get one and not the other. You're either going to get conditions of things like oxidative stress and DNA effects, which are concerns for regulators looking carcinogenicity, but you're also going to get cell death in those cases. You're going to lose those cells and you're going to get replenished with fresh and healthy and new cells. And that was what we saw, and that's what we think based on the simulation results. And so, at therapeutic doses, you're fine. There's minimal impact. There's no liver injury. There's no stress. You have ample antioxidant capacity to handle the acetaminophen that's being given and the effects that are there. When you switch to the overdose case, when someone starts taking 10 or 15 or 20 or 30 grams of acetaminophen, which does happen, unfortunately, from time to time as people take a whole bottle of the pills, that's when you start to see oxidative stress, DNA damage, but more importantly, liver cell death at the same time. And so we really don't see, based on our analyses, a zone, I would say, or a dose response and exposure response window where we would be concerned that you would be getting carcinogenicity. And, most importantly, I'll point out, it's recommended that patients take 3 to 4 grams of acetaminophen per day maximum. So the actual clinical, impact region here, the area of importance is way down to the left in terms of this exposure response. So interestingly, the FDA agreed with what we said, not directly. They didn't respond to us, but they did respond to OEHA independently of the CHPA, and they noted that we have determined that the currently available data do not support a conclusion that exposure to acetaminophen in FDA-regulated products causes cancer. They went on to say that a Proposition 65 cancer warning would misbrand these products in violation of the Federal Food, Drug, and Cosmetic Act, and therefore would be preempted under federal law. So this is a fairly strong FDA warning letter and communication, which is pretty public and went directly to OEHA and was posted up on the website. So there was a few things that I took away from this presentation, from this particular project that we did in collaboration with the CHPA and some of the sponsors there, that I think are important, that are general. They're not specific to acetaminophen or carcinogenicity as a use case. The first is that fit for purpose validation is very effective. It's often easier to defend. If you can validate your model in the context of your application, you're going to be able to win hearts and minds much more easily. And we have done this many times. So we've worked with compounds where sponsors have requested that we simulate similar compounds in the class, or compounds that were designed or intended for a similar indication, so that when they went to regulators, they could have confidence in the results because they have validation with a known entity. Or they've asked us to validate the model with a compound that's in-house, already been through the pipeline and maybe failed for liver injury purposes, and then simulate the backup compound. So they have some level of confidence that, yeah, you were able to recapitulate what we saw before, so now we think you can go on and do something new. And that fit for purpose validation is very powerful. It's especially powerful for those who aren't as familiar with modeling and simulation, and it helps communicate to them why they should believe what you're saying. The other thing is an integrated global response in general, where you take modeling results and you couple that with in vitro, preclinical, and in vivo results, is really powerful. And I think we all know that that's the idea behind putting together IND and NDA submissions with all this information together. But it just highlights how, as modeling and simulation experts, we can work harder to partner with other people, other groups, experimentalists and others, to make sure we're packaging things in a cohesive way. Next, publication in parallel with submission to regulators can provide and produce credibility and can be very useful in many cases. You've got that peer review happening, and sometimes it's hard to get sponsors to want to publish, but I think it's something that can benefit everyone. And then Lastly, a lot of times modeling and simulation tools can be used in a myriad of ways, and some of those ways are outside of the original intent or purpose. So with DILIsym, we used acetaminophen initially as sort of a building compound. We used it to build and validate some fundamental relationships in the model, like the number of hepatocytes and how that relates to liver enzymes in the bloodstream. We didn't intend to build DILIsym to specifically look at regulatory issues with acetaminophen, and yet we have done that now in a number of cases. So modeling tools are often useful for reasons that we don't realize until later. So with that, I appreciate everyone who's hung in there here at the very end of the very last day, and I'll be happy to take any questions if there are any.
Thank you very much, Brett Howell. Yeah, I got a couple of them we've got some time for. One was, so you've got some very nice data here with acetaminophen as a mono entity. And it's very much in line with, the daily recommendations of, 3 to 4 grams of acetaminophen at the most. But one of the questions we have is if you have any comparable data or information on combination products such as acetaminophen and the barbiturate butyl.
It's a great question. It's a timely question. We have been doing more and more, I'll call them DILI, DDI type simulation scenarios in our projects. And oftentimes we, in fact, I think 3 or 4 times we've had clients come to us and say, acetaminophen was given in our trial and we have some question about it. So can you run acetaminophen with our molecule? So we are doing that a lot lately. We've done it for compounds that have different mechanisms. Some of them cause oxidative stress that are similar to acetaminophen, some of them don't, and you just have different mechanisms all happening at the same time. But I don't have any specific examples of the compound you mentioned or that are published or public. But I will say we are doing quite a few DDI studies, and acetaminophen is part of some of those.
Obviously, you can't, confidentiality, et cetera. We have another good question that came in of, are you seeing an interest in using DILIsym for personalized medicine applications?
We're seeing a trend towards companies being willing to consider advice around what patients may be susceptible to an injury or an increased risk and how that would impact exclusion or inclusion criteria, or even advice for contradicted medications on the market. I think that we're probably one step removed from what I would call true precision or personalized medicine. We're probably more in the precision medicine, or sort of the generally speaking, can we qualify who's at risk, and can we better design our trials? Can we avoid these folks? But in terms of individual patients and saying, let me put the patient's parameters in that specifically are related to Bob, and then let me run the model for Bob and see what Bob would do well with. We aren't there yet, but I think that's something that down the road we can get to, across our different, PBPK and QSP and T capabilities.
Excellent. It's always good to see where the future could lead us. I have a question for the model validation. So validating your QST or QSP based model. Do you need to have the experimental data in 2 species when you're validating your model?
Not necessarily. I think it's again very much fit for purpose, and in a lot of cases, we are, as I mentioned, some of the cases where we have validation, we're actually just making a prediction of what happens in humans. And we're looking at clinical outcomes data that's related, for example, to liver injury. We don't have a lot of mechanistic data. We certainly wouldn't have in many of those cases, rodent data or preclinical data. So in this particular case, acetaminophen is a special molecule. It's something that has been studied for so many years, 50, 60, 70 years of literature on acetaminophen, and very, very detailed and mechanistic studies have been done in the field of toxicology on this drug for so long. So I would say we had an overabundance of data, not just in terms of across the species, but in terms of the number of studies and the depth of the studies per species. But I wouldn't say that you have to have more than one species to validate. It just depends on what's available and also what case you're trying to make. If it's a very, very dependent case, or if it's a case that's just supportive and you need something that gives someone some comfort that they should take some stock in what you're saying.
And I think that's in line with a lot of the themes we've heard from various speakers today of, a lot of times the questions you're asking is really going to drive what data you need going in and what types of models and the timing of those. So I think that's very much in line with what we've been hearing.
Yep.
I think with that, that brings us to the conclusion of this session. Thank you very much, Brett, for your time and everyone that joined us. As Lindsay posted in the chat, I wanted to remind everybody that we are going to have the replays and the presentations available. I'd say check our resource center next week for those start to be posted. And with that, I thank everyone for taking the time to join us. Have a wonderful day.
Yeah. Thank you, Ryan. And thanks everyone. Have a great day.
Thank you.
Bye.
Bye.